I'm Favalli Nicolas, a group leader working at Philochem in the chemistry team. Especially I'm working in the field of DNA-encoded chemical libraries, and Philochem is a pharmaceutical company located in Zurich, specialized in the discovery of small molecule therapeutics for the treatment of cancer. At Philochem, we typically use only DNA-encoded chemical libraries as a high-throughput screen methodology. At the beginning, we synthesize libraries containing between millions and billions of different compounds. Once we obtain the final library, we screen against a target of our interest and process that we call affinity selections. So typically from selection, we can identify even multiple hits. Sometimes we have also thousands of different hits. The main challenge is the resynthesis, because you have several hits that you would like to resynthesize, sometimes even thousands of compounds. Of course, you cannot resynthesize all of them, and the resynthesis sometimes also the off-DNA resynthesis can be cumbersome. The presence of DNA, of course, facilitates the resynthesis of these compounds, but once you have the DNA, sometimes you have a steric hindrance factor that sometimes interfere with your measurements. For example, with fluorescence polarization, sometimes we obtain dissociation constants that are a factor 10 or more higher compared with the values that we obtain with the same molecule without DNA. And with DNA, it's difficult to validate compounds, for example, by SPR. The DNA is a molecule that is negatively charged, so with the Biocore we face issues for the immobilization of the compound on the chip, or when we do the immobilization of the protein of the chip, we observe that the K-off are much faster because the DNA is not interacting well with the chip surface. Instead, with focal molography, we can exploit the presence of DNA for the annealing of the chip, and in this way we display the molecules, the hit candidates on the surface. Then we can flow the protein, and we can obtain directly kinetic parameters K-on and K-off directly on DNA. We're able to obtain kinetic profiles that we were not able to obtain by Biocore. Sometimes molecules that are not active by fluorescence polarization we were able to spot by focal molography, and machine is very user-friendly. We learn immediately how to use the machine. It's very easy because you just have to prepare your sample in different vials, the different aliquots with your target protein or with your DNA pool. You add the vials to the rack, and then the process is completely automated. For sure, we will add this methodology as an orthogonal methodology that will complement methodology that we have already internalized, for example, fluorescence polarization and ELISA.